National Marine Fisheries Service, who's in charge of monitoring and regulating populations of fish all up and down, all over the North America. And one of the things they regulate most closely is salmon populations. And so essentially what the folks in the salmon community have been doing, is the same thing we're trying to do with migratory birds, only they're way more funded because salmon are a commercial species, and so they generally kind of lead the field in this kind of stuff. So, and I always feel justified. My husband is actually a biologist for the National Marine Fisheries Service, and he developed this program called GSI-SIM for assigning birds back to their population of origin. And we then use the same program for identifying birds from specific migratory stopover points back to their population of origin. So this is what the resulting, we've been calling this a genoscape looks like. So if you've talked to Emily about her research, she deals a lot with ice escapes, and we're trying to one-up them here by coining a new word. It's all about genoscapes, not ice escapes, and to basically describe the spatial distribution of these genetically distinct populations across the breeding range. So again, this beautiful rainbow here at the top is the results of the structure analysis, again, this Bayesian assignment program. What you have here, let me just break it down, is you see there's actually a lot of little tiny lines all along this rainbow. Each tiny line represents the assignment probability of one individual to that particular population. The populations are indicated by colors. And then you have these letters here. Those are what I'm calling sort of a sampling site. And those letters correspond to the letters on the map here. So everybody in this A group here, there's about probably 15 individuals in this A group. They were all sampled from this location on the breeding range. And everybody in this A group had a very high, what you call posterior probability of being assigned back to the purple genetic group, okay? In cases where you have a lower posterior probability are where you get into sort of this group here, the Sierra Nevada, Pacific Northwest, and coastal California, where individuals, it's a little less certain whether they're from, say, the Pacific Northwest, or maybe in this case here, from coastal California. Okay, so the height of the bar, it represents sort of our certainty or our confidence in the assignment of those individuals to the distinct genetic groups. What I hope you can see is that we have very strong support, both from, again, so the colors here on the map are the results of GeneLand. So that's another assignment program that's, quote, spatially explicit, okay? So it's basically the same, two separate analyses, structure and GeneLand. In this case, GeneLand's actually putting the genetic groups onto a map. That's what I mean by spatially explicit. And both of the programs are essentially saying the same thing, okay? On the GeneLand results, the opaqueness of the color, so the more red it is, the more we are confident that birds from that region belong to a distinct genetic group. It fades here and becomes transparent because we have no confidence, we have no actual information about where birds from that area, which genetic group they belong to. Okay, so there's a lot of information in this map. But the sort of the general overview is, if you don't care about all the specifics, is that what the genome scan, the genoscape is telling us is that there's essentially 6 genetic groups: a Western Boreal, an Eastern, a Rocky Mountain, a Pacific Northwest, a Sierra Nevada, and a coastal California. And what's really exciting about this is that these genetic groups, particularly in the West, are actually on the spatial scale that birds are managed. So in the United States, there's something called bird conservation regions, in which basically the North America has been split up into different regions, and management strategies for particular birds are decided within a bird conservation region. And in the case of the West Coast here, these are almost identical with the boundaries of current bird conservation regions, which is really, really convenient for managers and people trying to develop strategies for these species. Okay, so then what we did, again, this is just the breeders. Then what we can do is take birds from migration stopover points. So each one of these arrows is representing a bird captured from a migration station, and the arrow is pointing north to represent these are birds captured during their spring migration when they're headed from their wintering grounds in Latin America to their northern breeding grounds. And then we assigned all the birds captured at these migration stations to one of the genetic groups, one of the populations of origin. And the bars sort of within the arrows represent the proportion of individuals en route to the different breeding populations, okay? Same thing for the wintering birds. These are birds captured at different times of the year. These represent, the pie diagrams represent the proportion of individuals headed back to the different breeding populations. So what you can see here is that birds that breed in coastal California, the Pacific Northwest, and the Sierra Nevadas, winter in southern Baja and western Mexico, and migrate north in the spring along a Pacific flyway route. Okay, that's really exciting. Everybody should cheer. Yay! That's so cool. Yeah, okay. And same here. So in the East Coast, we have eastern breeders found wintering in the Yucatan, Belize, and southern Costa Rica, and migrating along an eastern flyway. Then we have Rocky Mountain breeders wintering south of El Salvador and migrating, although the sample size is really low, they're migrating along the central flyway. And then we have this big blob of a group, the Western Boreal group, which is what Christina wishes was smaller and more informative, a collaborator of Emily and mine. And where we're not really sure what's going on here, but it's very widespread distribution everywhere apart from southern Baja and western Mexico, and found in all of the flyways from the central flyway all the way out west. Okay, so another super cool part about this work, and this was a part that I collaborated with some folks at the University of Southern Mississippi who were interested in stopover ecology of migratory birds. And those, their names were Christina Paxton and Frank Moore. This was Christina's part of her dissertation work. She actually sampled birds in a time series. So she was located at a migration station here in on the border of California and Arizona, and she was out there capturing birds every single day, capturing Wilson's warblers every single day, collecting information on feeding behavior and physiology of these birds. But in order to interpret her data, she needs to know, okay, well they're gathering fat at this particular rate. This is all a physiology associated with this particular migratory bird. But in order to understand why they're doing that, she needs to know how far are they migrating? Where are they actually going? Why do they need that much fat? What is the right environment at the stopover site? What's going to be best for these birds as you move across time? So what we did was to type the time series that she had, which was collected across 2 different years, 2008, 2009. And this is broken up by week. So what you can see here is that we not only have spatial structure in where birds are migrating, but we also have temporal structure. So you can see birds en route to coastal California are the first to come through that migration station, followed by birds headed to the Pacific Northwest, Then the Sierra Nevadas, the pink guys, show up here around April 12th. And then you have a distinct turnover between the week of April 26th and May 3rd, where the birds en route to the boreal forest region start arriving. So this is exciting to those of you who have ever been to a banding station or worked at a banding station and seen these waves of migrants coming in, and you want to understand more about where those migrants are actually going. This sort of data can really help give you that answer.
Are there size differences between the purples and the...
Yeah, that's a really good question. Christina would have that data. I actually have no idea. I just do the genetics. Yeah. But you would think, I mean, these guys are going farther. My guess is that they, they might have some sort of adaptations for having longer flight distances, like longer wings, but maybe more fat still left on. I don't actually know though. That's all just speculation. Yeah, so what I'm actively working with Christina on is to try and sort of refine this information so she can then develop sort of a better understanding of migrate ecology. Okay, so I'm doing a good job. I always think I have way too much time, and so I take a long time, and then it's like, oh my God, I only have 10 minutes. So I told you for about 45 minutes now all about how you would develop these tags and how they actually work for the Wilson's warbler. Hopefully I've convinced you that genomic tags or high-resolution molecular tags are a viable addition to bird banding for tracking migratory birds on a broad scale. And now, just in the last sort of 5 to 10 minutes, I just want to show you how the resulting information can be applied to inform conservation efforts. So one of the ways, this is all kind of areas of active research, but one of the ways that we're using this information is to begin to start to assess drivers of population trends at regional spatial scales. So one of the populations within the Wilson's warbler that's actually been declining precipitously since the 1970s is the population in the Sierra Nevadas. And so now that we have this information about the fact that the Sierra Nevada population is a distinct genetic group that is found wintering in western Mexico, we can really use that to refine our models of factors that may be driving population trends in that breeding range in the Sierras. So it's complex, and this is not my area of expertise doing the models, but luckily I work with a lot of fantastic collaborators that are taking this on. And what they're doing is to develop models that incorporate information from climate, they incorporate information from habitat changes, on all disease frequency in those areas, and put it all into a model to understand what are the main factors driving these distinct populations. I'll just briefly show you some of the results from one of those models that looked at the relationship between what's called the El Niño Southern Oscillation, or ENSO data, which is climate data, and survival in Wilson's warbler populations broken down by genetic group. So I apologize, this is preliminary data, and the colors do not match. So you might be wondering, oh, why? I thought Pacific Northwest was green. It is green. The green Pacific Northwest group was green, and Sierra Nevada group was actually pink. So anyway, when I come and talk next time, this will all be fixed up, and I'll show you the results of it. But for now, just the takeaway message is that we were able to divide the survival data by genetically distinct group, and then look at how climate oscillations may be explaining the survivorship that we see in these genetically distinct groups. Again, the main takeaway here is this red line is actually the El Niño Southern Oscillation. This blue line is actually survivorship trends for the coastal California genetic group. What this is saying is that there's an extremely tight correlation between climate and survival of populations that are found breeding in coastal California. And that climate, that relationship is much less so for other populations. So it seems like, at least for the coastal California breeders, we have a pretty good idea of what's driving population trends, but for the other ones we may have to do a little bit more digging.
But look at the Sierra one, it's a smaller population, but it's still doing.
Yeah, also tracking, yeah. Which color is Sierra?
Okay.
Sierra is green, yeah. Yeah, and same for purple, Pacific Northwest.
And this is all from banding data.
Yeah, this is from MAPS data. So the MAPS program regulates, is a network of banding stations. It stands for Monitoring Avian Productivity and Survivorship. And what they do is have a standard set of methods for recording data and estimating, so you can use the data in a coherent way to estimate survival. Yeah. So stay tuned for more. I have a call tomorrow with a bunch of great collaborators to, see about how we can take this data further. And then the other way that we've been actively taking this sort of work is to try and understand the impact of emerging renewable